You can do it on wool blood, but we did not have the time to test it with the protocols we have produced with you. The protocol has been optimized for PBMC, so we have prepared some fecal separation of mononuclear cells to do it.
Personally, I haven't seen protocols for whole blood. If there are some, maybe, but I don't know if they exist, so it's up to people to optimize.
Yeah.
As I know, other manufacturers, they recommend to use PBMC also.
Yeah.
Я говорю, что, насколько мне известно, нет производителей, которые рекомендуют цельную кровь использовать, поэтому лучше использовать PBMC. И, я просто знаю людей, которые в лаборатории пробовали из экономии времени покрасить цельную кровь, и да, вообще ничего не получается, куча входит специфического связывания. Видимо, это...
Не получается.
Получается?
Мы делали так.
С FOXP3?
Да.
Ну,
цельная кровь.
I'm not surprised because in the companies
может быть.
Много лет тому назад мы работали над этим.
They work a lot to prepare some kits, and they want the kits to be robust, not to have some complaints, so they stick to one protocol, but most of the times you can simplify it. They have a lot of washes also, we can see. We can probably simplify. And so you have done it on wool blood?
Yeah.
And how do you do erythrolysis? Do you do a specific erythrolysis or?
We did erythrolyze, but I can't remember the buffer's name.
Because as I was saying yesterday, maybe the permeabilization buffer that you use to permeabilize the cells, you destroy the membrane, and most probably you destroy the red blood cells also. So maybe you do not have to do erythrolysis, but I did not test it. We have done some FOXP3 labeling, and we always did it on PBMC. So you have to test it, and it can probably work. And then you can make it easier.
So if there is an established protocol, so you can follow it, but
you can make it simpler and spare some time and so on. Okay, but the permeabilization buffer, usually they don't tell us what is inside. It's probably from saponin, and it's the same thing that we use for cytokines, and it's very good to destroy the splitters and the red blood cells. So you see the small expression of CD25. It's very small, it's very close to the negative cells, and you can see that it's not expressed or almost not on CD8 T cells. Okay? So one application is that you can use CD8 T cells as a negative control to do your fusion, and then you do not have to do FMO or isotype control, because you have internal automatic internal controls. The other way also is that it's better. We will see. It's easier if you use this type of graph with the lines. I don't know what is the name. It's easier than with the dots. Because the line
contour plots.
Yes. You know what it is in Russian, yes?
А как по-русски будут графики такие, контурные, как контурные карты?
Чем больше этих полосочек, тем больше.
Тоже, наверное, I think contour is the same.
Oh yeah. Also, I know one Russian name. It helps a lot because this will give you the center of the second population and the first population. And there is, if you think about mountains map,
like on the map.
In the middle you have a valley, and it helps you to put the separation between the two populations. For example, I have also on Navios, and on Navios they do not have this type of graph, and it's not so easy to detect, to separate, to be sure about where to put your gate.
Navios, it's device from what?
Navios is Beckman Coulter instrument like Cantor.
Yeah, look on that plot, it's not always easy to know where you put your gate. This is an expression of Fox P3. Okay, and you see it's exactly the same type of pictures as you have here. It's not very strong, and it's on a very minority of cells. Okay, so you cannot expect to have a very strong positive and a strong negative.
Is it from your lab?
This is my, yes. Yeah. This was to show that CD25 positive or not.
Sorry, it's native blood impressions, or it's stimulated PBMC?
Oh no, it's native. But I think it was on fecal, I'm not sure. I am working in the hospital, and sometimes we have some clinicians asking us to detect T-Rex as a part of clinical study, and it's difficult to do FOXP3 on a routine basis. So most of the times we just do CD3, CD25.
Why they need to measure Treg?
Oh, it's most of the times it's a part of clinical studies. They want to see, for example, if there is immunodeficiencies in HIV, for example, or in transplantation or in some infection. So for the protocols, we have done fecal, we have washed the cells. You do the membrane labeling, CD3, CD4, CD25. You fix your cells, you permeabilize like a cytokine, and you go inside the nucleus to do the nucleus labeling of your FOXP3. It's the same, you have to try if you can go directly to this. You can try to do fixation, permeabilization, and then you can do membrane and cytoplasmic labeling. I did not test it, but might be also a way to simplify your protocol. And if instead of waking up at 6 o'clock to do your experiment, you can do it in 2 hours. It's what I said to my students, you have to be lazy, so you get more clever, you make some progress.
So after 2 months of optimization, you will be
you spend some time.
Spend some time, you can.
If it works. If it works. There is an alternative way to better detect your T-Rex is by using CD27 1 to 7. Okay, if you
why we use 1 to 7 marker,
on membrane.
but why we use it?
Look, because you have a much better separation between your FOXP3, your T-Rex, and the other cells. Okay, so here it's much easier to put your gates and to gate your cells and to count your cells. So because I am lazy, what we do on routine labs, on routine activity when they ask us to do labeling, to do the counting of the T-Rex, we do CD1 to 7 and 25 instead of doing FOXP3. Okay.
But best marker is FOXP.
This is the reference, yes.
Is there any critical difference between marking membrane CD127 and nuclear FOXP3? Do we have some critical difference?
There is a few papers showing that it's almost the same.
So today we're gonna try to check it. If our samples are good, we
have done all the labeling.
Done all the labeling. So if you look at your protocols, we have done both FOXP3 and both 25 и 127. И попробуем посмотреть. Но если FOXP3 хорошо получился. Потому что не всегда он...
After that, there is some teams who have demonstrated that there is 1% of Tregs that, but 1% of 1% of 1%. Maybe you can find that these cells are specific as extraction. But then you have to be very patient to have a very serious student. This is so an alternative and easier way to do it.
Also,
But it's not the classical way. So.
Also, it's the way if you want to sort, because FOXP3, when you stain, когда вы красите FOXP3, клетки
Your cells are dead.
It's all dead, поэтому если вы сортируете только 25, 25 и 127, if you sort.
Yeah. And you can only do it by flow cytometry because it's always faint. It's very low labeling of CD25. So you can just analyze it and say to a cytometer, sort these cells, because like magnetic cells, sorting it's impossible to do.
I just have a practical question. Did you sort this Tregs by surface markers and then also cultivate with, for example, PBMCs again with the antigenic stimulation? So did you see some differences without staining the cells and with staining? Do antibodies
interfere? I've not done it myself, okay? But the real way to prove that you have Tregs, inhibitory cells, is that to sort your cells, okay, as we have just described, and to do a T-cell assay and to see that you have a real inhibition. Okay, so this is the only way to prove that it is Tregs, because as I told you at the beginning, the definition of Tregs is that they down-regulate the immune response. Okay, you not necessarily can trust that FOXP3 is specific. So there is people doing sorting and doing down-regulation, and they don't bother about antibodies.
So they anyway inhibit
Yes, yeah.
After sorting.
Yeah. But they sort the T cells, that we have just discussed, with membrane labeling. So the way why I used this labeling, it was just to prove that the cells I identify as 25 and CD127 were Tregs, because I had to prove it for the paper, but I'm not doing it regularly. Sorry.
Is it possible to use the same markers for in mice to measure Treg in
Yes.
mice?
Yeah.
The same?
Yeah.
127 and
Yes, I think so. You can just a side remark. You can see that in peripheral blood, all your CD40 cells are positive for CD127. Okay. If you look in the same sample, if you look at CD8 T cells, you will have the part of CD8 T cells that will be negative. And if you use this type of graph, you can clearly see that there is population on CD8. Okay, and if you look at in different ages, in elder people, the population of CD8 here will go up, and this one go down, and you have a balance toward the CD127 negative CD8. Okay, so if you take blood from elderly people, you will have two populations in CD8, and you will start to have a second population of CD4. Okay, so it's a way to give an age to the CD, to the T cells, but it's not nothing to do with T.
So does it mean that elder people have more Tregs in blood?
No, because it's no, it doesn't change here. It's just the negative here. Okay. This population will move to the CD127 negative, because CD127 is the IL-7 receptor This means that it's expressed by naivety cells coming out of the thymus. In the thymus, they need IL-7 to proliferate, and they keep the receptor for sometimes outside, but after sometimes they become mature and they don't need any more IL-7. Okay? And if you we were discussing yesterday night with Bob Bagheras, and he was talking about studies in India and children who are living in very poor conditions. There are a lot of infections, things like that. And they do have some old lymphocytes, early aging of their immune system because it's too strongly activated. And we also have in HIV. In HIV, you have this population is going down because of the virus. Okay, not now with the treatment, but if the people do not use their treatment, this population is progressively destroyed by the virus, and the Tregs are not going down so fast. So the percentage of Tregs is going up. Okay, but they are not. I don't think in absolute number they are not going up, but in percentage they are going up because this population is going down first, and you also will have more old lymphocytes that will stay, and less young.
Can you explain once again what does it mean, these circles, geographical circles?
Oh, it's a statistic. You have a bulk of cells, and more cells you have, more you have a new cycle. Just like sand, if you put a sand, you have a mountain of the cells, okay, and you put a cycle to make a difference between the quantity of cells you have by circle.
Здесь клеток наибольшее количество, здесь меньше, здесь меньше, здесь меньше, меньше еще. Как контурная карта.
Is it clear?
Это не контурная карта, это называется топокарта. Топографические карты. Контурные, они как плоские, а топокарта, они как бы трехмерные.
We are organizing a summer school in Spain, and one night I was going to a beach. I have tried to put some to make some plot with sand, but it was very difficult to take pictures. So because it's a good way to understand the system. If you just do color dots, then you cannot make the difference between here where you have a lot of cells and here where you have few cells, because it's all uniform. Okay? If you have a density plot, you will have the dots, and more you have dots, you will change the colors, and you can see where it's
we can see it on the instrument, both type of plots.
But it's just tricky things that can help you to define your population. This was just an example. I've got some files we can analyze. So this is a normal patient who we can easily identify the Treg. Okay. Be very careful with dot plots, because sometimes you have dots here and they're on the axis and you don't see them. Okay? So be very careful, because you have to count these cells. They are real cells, but you don't see them. So each time you put a gate, try to put your gate above the axis. We can see very easily.
If you have a DIVA-based instrument, there is a feature you use by exponential scale.
Yes, this is the case.
Если у вас прибор, у которого программа DIVA, то там есть возможность экспоненциальное отображение, и вот здесь вы можете увидеть, что здесь below zero, то есть
You see there is a zero which is not possible with log scan.
Это математическая обработка, которая позволяет увидеть, если клетка здесь, то если 0 здесь, то она бы оказалась на оси. So I'm explaining that if the
Yes.
cell is here, then if 0 is here, then
Yeah.
everyone will be on the axis. И это очень удобно, потому что визуально очень тяжело, когда клетки лежат на оси, их вообще не видно.
So this is very easy to do. You just have to think. We will show you, and
Еще
think of using this technique because
So,
it's helpful not to make mistake.
It is possible in Diva and it is possible in FlowJo.
Yes,
But
in most
I don't know in other.
Yes, in Kahoot!, it's okay.
others it's not possible.
Yes,
I
it
know,
is.
But
Yeah,
is it?
yeah. In most of the software it is possible, and it's just a matter of graph, and it doesn't change anything about statistics. Oh yes, so this is a normal expression of Tregs only by membrane labeling, okay? And we had a problem with some patient who had low Tregs, and this was kidney transplant patient. So the clinician was asking why, in fact, it was not a very good question. The question was that at some times we have tried to use monoclonal antibody against CD25 to cure the rejection. Okay? The idea is that you graft a kidney, then you have an acute activation of cells because of the allograft. Okay? And their idea was that if you put antibodies against CD25, you will kill the specific cells that have been activated by the graft, and then you will induce a tolerance. It didn't work. It didn't work, and sometimes they wanted to check if there is still Tregs, because the risk was that you can have a inverse effect if you if you destroy all the Tregs. And we did the analysis on cytometer, and just by doing membrane labeling, we could not detect any Tregs. Okay. So here we have to do FOXP3. We have done FOXP3, and we could show that there was still Tregs that did express CD3, CD4, FOXP3, but we could not detect CD25 because the patient was treated with monoclonal antibody anti-CD25.
I think I should explain, because это довольно сложно. Может быть, я переведу. Но суть в том, что была идея у врачей в центре, где работает Клод, что если при трансплантации аллогенной почки, то есть от другого человека, если дать препарат моноклональных антител против CD25, то тогда элиминируются все клетки, которые будут отвечать на этот трансплантант, и реакция как бы исчезнет. Но этого не произошло клинического эффекта. So there was the clinical effect. Did I got right?
Not strong enough, and so they stopped to use this technique.
То есть реально клинического эффекта не произошло, но они решили еще посмотреть, что происходит с терегами, убиваются ли тереги при этом.
They didn't want to induce autoimmunity or some other side effects.
So, да, они не хотели, чтобы это приводило к аутоиммунному процессу. Вот, и как бы как часть исследований было посмотреть тереги. И интересно, что вот эта картинка оттуда, то есть CD25 клеток не было, но когда покрасили FOXP3, am I right that when you stain FOXP3, you have managed to see the Tregs? То есть по FOXP3 были положительные клетки, несмотря на вот такую картинку.
So the next question was to know if the CD25 was internalized, or if it was just a problem of immunostaining.
And how did you resolve this?
So we bought 12 anti-25 antibodies, and we found that we could detect the CD25 on the surface of the cells with some clones, only 2 clones, and the other clones were the could not recognize the protein.
So
So it was just the epitope was masked.
So you did detect 25, but with other clones, right?
Yes.
Ну, то есть они испробовали 12 разных клонов, которые против CD25, которые к разным эпитопам. И two of them, да?
Yes.
Two of them, they, у них получилось обнаружить. То есть получается, did we get right that there was CD25 on the surface, but the epitopes of the which our antibodies do stain, they were just blocked
By the treatment?
by the treatment.
Yes. And
Да,
if
то
we
есть
use
получилось, что
another staining, it was working. There was one antibody from PD, and we have tested 12 antibodies. I can give you the paper, I don't have the details of the name of the clones, but this means that the treatment that is used for the therapy was covering one site of the molecule. Okay, and if you find a clone that recognizes another site of a molecule, you could still detect the molecule. So the molecule was still outside, but the site was just covered by.
it's very interesting because I draw more broad conclusion that if the antibody, therapeutic antibody binds to cell, it doesn't mean that the cell is killed, because if you see the cell, you detect with other antibodies, the cell is living, right? It's functioning probably.
Yeah.
So that your antibody just doesn't work, but it's very, very interesting because it's to produce it costs millions of, I don't know, dollars, and to labor of huge companies.
It has nothing to do with T-Rex, but we had the same question with treatment with anti-CD20 for B cells, rituximab and various new one. Now it's very large, widely used for different diseases like autoimmunity, and the people were asking us if there is still some B cells left or not. Now they ask us to detect how low we can detect B cells to see if their treatment is efficient or not, just like minimal residual disease veto for leukemia. And the question was to know if we could not detect B cells, or it was very, very, very low. And their question was to know if we were sure that it was not because the CD20 was masked, or if it is there was no cells. This was easier because we used CD19, which do label another protein, and we could not see any B cells. Okay.
Понимаете, что произошло? То есть получается, что в данном случае просто вот эти терапевтические антитела не закрыли сайты все эпитопы на молекуле своей, на молекуле CD25, но клетка осталась живая при этом. И
Но она не окрашена.
она не окрашивается, потому что это место уже занято, да. Но, да. Да, их нету, да. Да, и получается, что как бы я так понимаю, что тот, кто эти антитела разработал, он планировал, чтобы клетки умирали от того, что то есть он планировал убрать все 25 клетки, они планировали, а получается, что они так и плавают, покрытые этими антителами, и с ними ничего не происходит.
Но если они конъюгированные с хлорохромом, как не...
Это терапия, то есть просто немеченные, чтобы, как ретуксимаб.
And then you have a redistribution of the B cells.
Yeah, in rituximab,
Yes.
I know there are a lot of generics now.
Yes,
which,
and it's very efficient for autoimmunity if you want to stop the production of antibodies. Or if you have lymphoproliferative disease and you want to kill
I mean, it is some people who develop these antibodies can draw conclusions that if it binds, it doesn't guarantee them that
No.
the cell is killed.
And you not always know what is the mechanism. So this is the genetic disease, which is called IPEX syndrome. This is a genetic mutation on the FOXP3 protein. Okay, so the babies do not produce any Tregs, and they do produce some autoimmune disease that usually you only see in a few people in adults, and they have frequently thrombopenia with no overcoverage. They have frequently thyroiditis, which